Welcome back. Welcome back here in the studio. I'm very happy that you're all still joining us. Between 500 and 600 people have been joining us during the day, and we hope that you stay with us because we really love to interact with you through Slido and everything. But for now, maybe the camera can show you that I am surrounded by masked people, mystery guests. in these strange times, we need to take these actions and protect everyone. It's the rules also, so everyone's wearing a mouth mask until they stand still or sit.
Yes.
And with me is Priyanka Rao.
Hi.
You are able to lose the mask already because you're sitting down.
Yeah.
Welcome. It's lovely that you are here. You are a postdoctoral researcher and you are a team leader at the Molecular and Cellular Neurobiology Department.
Yeah.
And you're also kind of the captain of the Pecha Kucha team,
yes,
right?
That's correct. Yes.
Because that is what we'll be doing with these mystery guests here. And if they take off their mask, they'll reveal how pretty they really are, right? Or handsome. The Pecha Kucha segment. What is Pecha Kucha?
Yeah, thanks for having us. So the Pecha Kucha segment basically consists of each presenter having 20 slides, and they have 20 seconds of commentary per slide. Importantly, the slides are all automatically paced, so they've really practiced, they've put in a ton of hard work, and they will give us a great overview of the research that they have been doing.
And why is this so appropriate for scientific results?
I think for the audience, it's a really nice way to get a really fun and fast overview of the research that has been carried out in all the different programs of Amsterdam Neuroscience. For the presenters, besides learning to deal with stress and rapid heartbeats, they also learn, I think, how to distill information to give their main message of their research in this way.
Yes, I can see that. And who are joining us today? What kind of people do we have here?
Yeah, we have researchers from all the different programs of Amsterdam Neuroscience across the different institutes.
Yes.
I think most of them are either PhD students or postdoctoral researchers.
From all different areas and departments.
Yeah.
Okay. And why are they here and not other researchers?
So they have been selected by the program leaders. So they've been picked because they've been doing such great work.
I see.
And we want to showcase the work that they have been doing in the past years.
They've been carefully selected.
Yes.
All right.
Correct.
Is there anything more to say before we...
Yeah, I would like to introduce the speakers.
Yes, please.
So we have a number of speakers here today from different programs. We have Esther Visser, and then we have Shu from complex trait genetics. We have Kitty Reemst from mood, anxiety, psychosis, stress, and sleep. Then we have Dirk Jan over there, who is from brain imaging. We have Hanne Lammertse from cellular and molecular mechanisms. We have Naomi Buntsma from neurovascular disorders. Then we will be joined via Zoom by Anke, who represents neurodegeneration. And finally, we have Broeders, who is from neuroinfection and inflammation.
Yes. Well, it probably will be quite interesting, but we also have a winner to pick.
Yes.
So what we will do, because we want to know who you think performed the best Pecha Kucha, what was the best Pecha Kucha, the criteria are up to you to decide. What you do is you go to Slido. And you will see in the polls section that the names of the presenters will be there. And we ask you to vote later on, and we will not show the results yet. And then in the end, the winner will be revealed.
Yes.
Yeah, that will be quite exciting, right? So I think you have the power to start the segment. So if you think that everybody's ready, you can say first presenter.
Yes, let's start with the first presenter, Esther.
All right, good afternoon, everyone. My name is Esther Visser, and I just started the final year of my PhD in the Memory Circuits team at the Department of Molecular and Cellular Neurobiology at the VU. Here we investigate how persistent neuronal ensembles can drive conditioned alcohol seeking and consumption. And today I'll tell you about this. So on this slide, you see 380 tiny little humans that represent the 380 million people worldwide that suffer from alcohol use disorder. Alcohol use disorder is a chronic psychopathology that's characterized by the persistent use of alcohol and frequent relapses that can occur even after long periods of abstinence. These kind of relapses can be evoked by alcohol-associated stimuli, such as bars or alcohol commercials, for example. These kind of stimuli evoke activation of alcohol-activated memories and can thereby induce strong feelings of craving. But where does the brain store these type of alcohol cue memories? Well, human imaging studies have shown that the medial prefrontal cortex, or MPFC in short, becomes highly activated in response to alcohol stimuli. As you can see in this graph, the MPFC becomes highly activated in people that have relapsed back into alcohol consumption, both compared to controls and compared to people that remained abstinent successfully. In our lab, we use an animal model of alcohol use disorder, in which we pair administration of an alcohol reward with the presentation of a cue light. Even after long abstinence, only presentation of the cue light can evoke relapse. If you lesion the PFC, this relapse is strongly reduced. However, rather than being a phenomenon of brain region-wide activity, we now know that expression of a memory is actually hosted by only a small number of distributed neurons, so-called neuronal ensembles. And as you can see in this picture. And as we know that the medial prefrontal cortex is very important in long-term memories, we hypothesize that neuronal ensembles in the PFC that are activated during cue-paired alcohol self-administration may support a long-lasting alcohol cue memory and can thereby drive relapse later on. But first, let's make an alcohol memory. So for this, an animal is put into an operant box where it learns to press a lever, which will lead to an alcohol reward and the presentation of a cue light. Our working model is that the medial prefrontal cortex ensemble that's activated will actually encode the association between the reward, the cue, and the context. But how do we target these MPFC ensembles? For this, we use our newly developed viral trap method. This method allows us to activity-dependently label these ensemble cells either with a control protein or with a DREAD, which is a protein that will allow suppression of these cells by injecting its ligand CNO. But first, we have to make sure that these animals are habituated to the taste and effects of alcohol. For this, they get 4 weeks of access to a bottle of alcohol and a bottle of water. As you can see in the graph over here, animals strongly prefer alcohol over water. This strong preference we see in about 90 to 95% of mice. Then, animals have to learn the task to lever press to obtain the alcohol reward. As you can see, over the sessions, animals make more lever presses, indicating that they have actually learned the task. Cells that are activated during this last alcohol self-administration session will be tagged. And then 1 month later, animals are again put back into the alcohol self-administration box, but now lever pressing does not lead to an alcohol reward nor to the cue. As you can see in the right graph, actually this reduces their lever pressing substantially. Now we have extinguished alcohol seeking evoked by the context, we can look at the cue. When we expose animals again to the alcohol-paired cue, in absence of alcohol, we see that in control animals this indeed evokes relapse to alcohol seeking, and that this is significantly reduced in animals where the ensemble was suppressed, as you can see in the black bars over here. To actually show that this is specific to alcohol activated neurons and not just any MPFC ensemble, what we did is we trained animals similarly, but now we put them into a novel context on the day of tagging to label novel context activated neurons. And here we don't see an effect on relapse to alcohol seeking. What follows from this is also that you would expect reactivation of the alcohol self-administration tagged ensemble, but not the novel context ensemble. So what we did is after tagging, we waited 1 month and put the animals to a cue-induced relapse test. And indeed, what we observed is reactivation of the alcohol ensemble, but not of the novel context ensemble. Then we wanted to see if animals if this association also holds true for other types of rewards, such as sucrose. Therefore, we trained animals to lever press for sucrose, but suppression of this sucrose-activated ensemble did not suppress relapse to sucrose seeking. So from this, we have seen that cells in the medial prefrontal cortex that are activated during alcohol self-administration form a long-term substrate mediating cue-induced alcohol seeking after long abstinence. But what about actual alcohol consumption? Is this also mediated by MPFC ensembles? Well, this is something we're now looking into. For this, we put animals in their home cage and expose them to alcohol and label cells that are activated during alcohol drinking. Then, a month later, we assess alcohol drinking first in absence of ensemble suppression and then with ensemble suppression. As you can see, this indeed reduces alcohol drinking as well. Interestingly, when we put animals into a different context, we don't see this effect on suppression of alcohol drinking. So all in all, I hope by now I have convinced you that relapse to alcohol seeking and consumption is mediated by a persistent cortical memory trace. Even though these ensemble cells are very small in number, they strongly influence behavior. And our next step is to find out whether there are molecular and physiological changes that are unique to these ensemble cells. And with this, I would like to thank my supervisors, Michel van den Oever and Guus Smit, my awesome colleagues here on the screen now, and all of you for your attention.
Thank you, Esther. Next, we have Shu Liu from Complex Trait Genetics.
Hello everyone, my name is Shu. My research interest is to combine the genetic information and the neuroimaging information to find the possible underlying neurological maximums. Here, the title of my report is Distinct Relationships of Genetic Risk for Mental Health and Cognition with Brain Structure and Function. First, we need to know what is the genome-wide association studies, also called GWAS. Human DNA contains millions of SNPs. Their variations in the DNA sequences can affect people how to develop a disease, cognition, behavior. GWAS aim to discover SNPs associated with a disease or a continuous traits. Previous GWAS studies have found that mental health and cognition had a polygenic architecture. For example, the schizophrenia is not controlled just by a single gene, but rather by multiple genes that each make a small contribution to the overall outcome. And based on the GWAS summary statistics, we can calculate the polygenic score. Here is an example. We can get the genetic effects as estimated in GWAS. We can also know how many risky allele in each SNP. Then we can calculate a weighted sum. This is the polygenic score. And the effects of the genes are not directly expressed at the level of behavior, but rather are mediated by the molecular and circular effects on the information processing on the brain. So the brain structure and the function work like a bridge, link the genetic variation to various traits. Using MRI, we can show the brain structure. The cortex mainly contains the gray matter and the white matter. White matter is organized into the tracts of axons, and the gray matter contains most of the brain's neural cell bodies. The gray matter is found to be involved with the sensory perceptions. For example, the seeing, hearing, decision-making, self-control. So we focus on the gray matter. People always use the cortical surface area and the thickness to measure the brain gray matter structure. And we can also use functional MRI to estimate the brain activity by detecting the changes associated with the blood flow. As we can see, a region in the brain is in use. The blood flow to that region also increases. Then we can get 4D fMRI data. Next, we need to extract the time series of different regions. We can calculate the amplitudes of low-frequency fluctuations for a given region. We can also calculate the correlations of time series between two different regions. This is the functional collectivity. This study systematically investigates the associations between the polygenic scores and the brain structure and function. And the data sets include about 36 individuals with both genetic and neuroimaging data. Then we do the Pearson correlation analysis and FDR multiple testing correction. For the total cortical surface area, we can see the polygenic scores for ADHD, smoking initiation, and cognition show highly significant associations. And in contrast, polygenic scores for most psychiatric disorders are not significantly associated with the total cortical surface area. And for the average cortical thickness, no significant associations are found. In fact, during the evolution of the primates, the cortical surface area has increased enormously, but the cortical thickness has remained relatively constant. Next is the functional measures. The whole brain is divided into 21 networks. We investigate the associations between the polygenic scores and the fluctuation amplitudes within these networks. Widespread significant associations are found with the polygenic scores for psychiatric disorders and educational attainment. For the functional collectivities, we can see the polygenic scores for psychiatric disorders show widespread significant associations, especially for the schizophrenia, major depression, bipolar, autism, ADHD. And the polygenic scores for cognition also show widespread significant associations with the functional collectivities, especially for the educational attainment. But very few significant associations are found with the polygenic scores for substance use. We can see the polygenic scores for cognition and ADHD are significantly associated with both brain structure and function, and the polygenic scores for psychiatric disorders show stronger associations with brain function than brain structure, and the polygenic score for substance use is only significant with brain structure. Their relationships are complex and distinct in order to decompose the association pattern of various traits with brain structure and function. We perform the partial least squares regression. The input data can be decomposed into genetic components and phenotype components. The first genetic component, the polygenic scores for ADHD, smoking initiation, and cognition are selected. And the first phenotype components, only structural measures are selected, especially for the cortical surface area in the frontal and the temporal regions. The second genetic component, only polygenic scores for psychiatric disorders are selected, especially for the autism, bipolar, schizophrenia, and the major depression. And the second phenotype component, only functional measures are selected. Then we can get the conclusions. The polygenic scores for ADHD and cognition show stronger associations with brain structure and function, and the polygenic scores for psychiatric disorders show stronger associations with brain function than brain structure. This is my email address. If you have any questions, you can contact me. Thank you.
Thank you, Shu. Next up, we have Kitty Reemst from Mood, Anxieties and Psychosis, Stress and Sleep.
Hi everybody, my name is Kitty. I'm a PhD student at the UvA Sils and in the lab of Anniko Korosi and Paul Lucassen, speaking today indeed for MAPS, Mood, Anxiety, Psychosis, Stress and Sleep. And today I will discuss with you how early life stress and diet can shape our central immune response for life. So early life stress is known to increase the vulnerability to develop cognitive impairments and psychopathology later in life. And in our lab we study preclinically how early life stress might affect brain and behavior. And we use a mouse model for that. And in the next slide, we see that we give the dam very limited bedding and nesting material during the first week of life. And this leads to stressed dams and pups. And in adulthood, we see that the offspring develops cognitive impairments, changes in hippocampal neurogenesis, but also changes in the neuroimmune system. We have recently published that a dietary intervention with increased omega-3 fatty acid availability, which also has anti-inflammatory capacities, can prevent the early life stress-induced cognitive impairments. Here shown with the Morris water maze, but this is also true for different cognitive mouse tests. Okay, next slide. I thought it was going faster. It's stuck. Okay. Okay, yeah. So we also know that early life stress leads to a reduction in the survival of hippocampal adult newborn cells, and that this can be prevented by the dietary intervention with this increased omega-3 availability, and also that the early life stress-induced increase in CD68 positive microglia can also be prevented, which is a marker for phagocytosis. So since this diet was so potent, this led to the following research question for one of the following up experiments. Do early life stress and dietary omega-3 availability affect the inflammatory response to a later life immune challenge? I think there's something wrong with the timing of the slides.
It's helping now, but maybe we can
I will
do something to get it started again.
Okay. Should I just... Should I continue?
Yes.
Yeah, okay. So during the first week of life, we have control our early life stress. Then we give a standard or improved omega-3 diet. I will go further. So we looked at plasma cytokines, which were increased by LPS. And we also saw that early life stress can increase both basal and LPS-induced levels of 3 cytokines, which means that early life stress can indeed also modulate long-term the peripheral immune system.
Next one, just say so.
Oh, okay, next one. Sorry. Next one. I'm sorry.
Next one?
Yeah. The research questions for the hippocampal microarray dataset were: does early life stress or the early dietary omega-3 availability affect gene expression under basal conditions? But also, does early life stress and the early omega-3 fatty acid availability affect gene expression under immune challenged conditions? I didn't explain the setup very long this time. So one way we analyze this data set is using a weighted gene co-expression network analysis, which is an unsupervised approach to find structure of co-expression in your data. And the output are the so-called modules, which are sets of genes of which the expression is highly correlated. And then when we find these modules, we try to see whether they correlate with our predictor variables. In our case, either the stress or a diet or the LPS. And to find more biological information about these genes, we also perform a gene ontology analysis on the genes within such modules to get a better understanding of what they might do. Next slide. The first module that we found, the largest module, was correlated to early life stress and consisted of genes that are related to structural components of the synapse, which is interesting considering our early life stress-induced learning deficits. And then the second module that we found, next slide.
Wait, because if you talk too fast, then...
Oh, but it's going automatically again? Okay, sorry, I didn't know. Okay, so this module was consisted of genes that are related to the extracellular matrix and transmembrane transporters, which is also interesting considering the fact that these fatty acids can be incorporated in membranes and have all kinds of downstream functions. So, short recap. Under basal circumstances, we see that early life stress can affect structural components of the synapse, which is interesting for us because of these early life stress-induced learning and memory deficits. But also the module related to the diet gives us new ideas on how this diet might be working in the brain and might affect cellular communication, for example. So we know a little bit more about what happens under basal circumstances, but what happens under immune challenged conditions. So we injected animals with LPS. I think I didn't say that before. And we see here a clear separation in gene expression. But what happens, what do early life stress and the diet do? First of all, when we compare LPS-injected control and early life stress animals that were fed the standard diet, we see that there are differences in genes related to the immune response. So in short, yes, early life stress modulates hippocampal gene expression under these immune-challenged conditions. Then, when we look more into this early life stress modulation of gene expression in these LPS-exposed animals, we see that this is actually dependent on the early diet. So the gene sets that were altered between control and early life stress animals fed a standard diet were absent or different in animals that were fed the improved omega-3 fatty acid diet. And we can appreciate this diet effect even more when we compare saline to LPS animals. And then both in control and early life stress animals, we see that the gene expression response to LPS depends greatly on the diet that the animals were fed early in life. You can see this in the outer circles of the Venn diagrams. So under basal state, we believe that early life stress affects synapses and the diet could affect cellular communication. And both of these mechanisms could be involved in the early life stress-induced deficits and the diet-mediated rescue of learning and memory. Under immune challenge conditions, we see that depending on availability of early dietary fatty acids, early life stress modulates the expression of immunomodulatory gene sets. But that also, when we compare saline to LPS animals, we see that the diet has a strong effect on the response to LPS, both in control and stress animals. Thank you for your attention.
Thank you, Kitty. Next up, we have Dirk Jan from Brain Imaging.
Hello, everyone. My name is Dirk Jan Ardesch, and today I'd like to talk about human brain evolution. But first, let's take a moment to think about the bizarre behavior that we're all currently taking part in. You're looking at a computer screen, either at home or in the office, and yet you can somehow see and hear me talk, even though I may be tens of kilometers away. These unique behaviors in terms of tool use and communication are, of course, made possible by our brains. What is it then that makes our brains so special? Well, this is of course a fascinating question, but it's also a challenging one, because we tend to look at our brains from our own human perspective. And just like the brain is the most important organ we have according to the brain, it's tempting to think that we also have the most unique brain in evolution. So, how much of a special snowflake are we then really? And more importantly, which brain adaptations make this possible? Well, we know already since a long time that the human brain is much larger than expected from a typical primate of the same body size. And this is due to a period of rapid brain expansion in the last 2 million years. But did the brain just blow up like a balloon? Or did certain parts grow faster than others? Well, let's take a look at the reconstruction of the human cortex. You should be able to see that it's much more convoluted, and that's because it has more surface area. And also, if you compare it to the chimpanzee, the prefrontal cortex is a lot bigger. And this is often suggested as a human unique feature. However, there's been lots of discussions about whether we can actually make such claims about human uniqueness by only comparing a few species. And this comes down to the concept of allometric scaling, which means that as a certain part of the body grows, another part grows in a different tempo. And in the following example of the skeletons of a cat and an elephant, you can see that the elephant is not simply a blown-up version of the cat with some added tusks, but you can see that the bones are proportionally much thicker. Similarly, in the brain, some parts of the brain may scale faster or slower as a general function of brain size. So, if we want to establish what are the expectations in terms of scaling of certain structures of the brain, we need to establish a trend line. And for this, we need a decent number of primate species for which brain imaging data is available. Unfortunately, this type of data is still quite rare. So, together with the Primate Brain Bank here in Amsterdam and the University of Oxford, we acquired high-quality neuroimaging data for 7 non-human primate species. These brains were donated by zoos across the Netherlands after the animal had died of natural causes. So no animals were sacrificed for this research. We then combined this data with already existing data to arrive at a total of 14 species, together covering more than a 350-fold difference in size from the smallest to the largest brain. Now we were ready to ask whether certain parts of the brain were specifically enlarged in humans. So let's take a look. In this figure, brain volume is plotted on the horizontal axis, and cortical surface area is plotted on the vertical axis. And this gray line denotes the expected scaling if the brain was just blowing up like a balloon, since surface area scales to the 2/3 power of volume. If we then add our primate species, we can see that the line of the primate brain is above that 2/3 line. This means that larger brains have proportionally more surface area. What's maybe also interesting is that the human brain, the red dot, fits perfectly on this primate trend. And this indicates that the amount of cortical surface that we have is actually typical for a primate of our brain size. However, if we then look at the corpus callosum, the main tract connecting the left and the right hemispheres, we can see that it does not keep pace with cortical surface area. So even though the human brain has the largest corpus callosum surface in absolute numbers, it is relatively much smaller. And this is very interesting if you think about it, because a larger cortical surface can hold more neurons, which would then need disproportionately more axons through the white matter to stay equally connected to each other. The fact that we instead see a relatively smaller corpus callosum indicates some kind of connectivity bottleneck in larger brains. Well, at the Connectome Lab, we are of course very interested in connectivity, so now I would like to share some of the implications for the connectome. Theories have already suggested a few decades ago that as the brain grows, it will be more difficult for distant regions to stay connected to each other. So the network of white matter connections will become more modular, as shown in this figure. So in this figure you can see that. And now we finally have the data and the techniques to answer or to confirm these old theories. And indeed, if we compare the human and the chimpanzee connectome, we can see that the human is more modular. Especially the connections running between primary sensory motor regions between the left and the right hemispheres are weaker in humans. And this may make the two hemispheres more independent of each other. However, the brain may actually compensate for a loss in interhemispheric connectivity. And this figure shows that across a wide range of mammals, the less connectivity there is between hemispheres, the more connectivity there is within hemispheres. And this combination of more connectivity within hemispheres, together with increased modularity, could eventually lead to unique specializations in larger brains. Here we find that connections between Broca's area and other language-related areas are stronger in humans, whereas connections to the remaining cortical areas were actually weaker. And this indeed points to some kind of potential language specialization in humans. So then, can we also predict these white matter adaptations from a general primate trend? Or are these patterns of brain wiring what actually makes us special? Well, with the data and the techniques that we now have, we can finally start searching for answers to those questions.
Thank you, Dirk Jan. Next up, we have Hanne Lammertse from Cellular and Molecular Mechanisms.
Hello everyone, my name is Hanne Lammertse and I'm a PhD student in the lab of Matthijs Verhagen. During my PhD, I worked on a neurodevelopmental disorder known as STX-BP1 syndrome. But first, what is a neurodevelopmental disorder? Well, brain development is a very unique and highly regulated process, during which a small clump of embryonic cells gradually grows out towards a fully functioning adult human brain. Sometimes something goes wrong in this process, and this leads to a neurodevelopmental disorder. There are many things that could cause this, but today I want to focus on genetic mutations that alter neuronal communication. The adult brain comprises billions of neurons that interconnect to form a network, and that network supports all the main functions of the brain, all the way from breathing and movement towards perception and thought. So it's essential that communication between neurons runs smoothly, and this happens at contact sites known as synapses. At synapses, vesicles filled with neurotransmitter molecules are released to convey a signal from one neuron to the next. Understandably, this process needs to be extremely reliable. And so at the synapse, there is a group of highly conserved proteins that together orchestrate this release of vesicles into the synapse. Together, they are called the snare machinery. Given the importance of this process, we can imagine that if something changes in the snare machinery, that could have profound consequences. And indeed, mutations in snare genes are associated with symptoms such as developmental delay, epilepsy, and neuropsychiatric problems. Collectively, we call these disorders snareopathies. Out of the snareopathy genes, the most commonly affected one is STXBP1, encoding a protein also known as MANKETIN1. This protein is absolutely essential for synaptic transmission. If we take away this protein completely, there is no synaptic communication happening in the brain. Patients with STXBP1 syndrome tend to carry a mutation in one of their two genes. There are point mutations across the entire gene, deletions as well as truncations associated with STXBP1 syndrome. It is important to realize that the type of mutation does not correlate with the severity or the type of symptoms. So genotype does not correlate to phenotype. If we make a mutant version of STXBP1 protein and we express it in cell lines or in mouse neurons, we see that the total level of this protein in these cells is much lower than if we express a wild-type protein. This suggests that disease mutations are negatively changing the stability of STXBP1 protein. We can mimic this situation essentially by making a heterozygous mouse model that has only half the usual amount of STXBP1 protein in their brains. And interestingly, these mice show cognitive deficits, epileptic seizures, as is shown on this video, and abnormalities in their brainwave patterns as measured by EEG. So, taken together, we think that STXBP1 disease-associated mutations cause a breakdown of the mutant protein, leading to a situation where there's not enough MANKETIN at the synapse to support its crucial role in vesicle release. We think this leads to changes in brain activity patterns and thus cause symptoms, but we do not actually know how that happens. And that is what we set out to find out. We created a multi-level study into STXBP1 syndrome, where we first look at the clinical profile of these patients, then we analyze brain activity patterns by EEG, and thirdly, we look at the cellular molecular level in a disease-relevant cell type, human neurons. To start out with this, we organized so-called patient clinic days, where we invited groups of patient families to Amsterdam to participate in our research. We aim to bring together patients, their families, clinicians and researchers from different fields of expertise, so that together we can start to understand STXBP1 syndrome at all levels. Firstly, we made a clinical profile for each participating patient, using interviews and examinations by the physicians and by a questionnaire that is filled in by the parents. Since then, we have started to reach out to physicians all across the world with the aim of setting up a worldwide STXBP1 clinical cohort. Secondly, we gathered EEG recordings from our patients. In a clinical setting, EEG is usually analyzed in terms of abnormalities in the EEG signal, as is illustrated on the top. However, we are instead looking at the normal-looking signal in between, or the resting state. By looking at this resting state, we can derive many different neurophysiological parameters. And by integrating these parameters, we look for biomarkers or characteristic features that may distinguish STXBP1 patients from others. Moreover, these biomarkers are indicators of changed brain activity patterns, and thus they may inform us about underlying disease mechanisms in the brain. Thirdly, we want to understand STXBP1 mutations at the cellular and molecular level. Previously, I already showed you some of the work that has been done in model organisms, such as mice. But arguably, if we want to understand the human disease, we should be looking at the most disease-relevant cell type there is, in this case, human neurons. And we can make those in the lab. By taking a skin biopsy from our patient, culturing the skin cells, we can reprogram these into induced pluripotent stem cells, which are capable of forming all cell types in the human body. From there, we differentiate them into human neurons that now have the exact genetic makeup of the person who donated the skin cells to us. Currently, within our project, we have samples from 7 STXBP1 syndrome patients and from 3 control individuals. And we're using a range of assays to study these. For example, we look at morphology, we look at their expression profile, but we also look at their functionality by studying synaptic transmission both in isolated neurons as well as in networks using live imaging. Having studied each of these levels at their own merit, we now want to integrate and combine this information so that we can understand how a single genetic mutation eventually leads to symptoms in patients. We believe this will not only help us to understand STX-BP1 syndrome, but also other neuropathies and perhaps even more broadly other neurodevelopmental disorders. That brings me to the end of my talk. I have many people to thank, most of all Annemiek van Berkel, with whom I set up this study, and Simon Houtman, who has become the EEG expert for STX-BP1. But finally, I want to take a moment to thank the patients and their families, who have been so enthusiastic and very willing to help us out and contribute to our research. Thank you.
Thank you, Hanne. Next, we have Naomi Buntsma from Neurovascular Disorders.
Today I have the honor to present you something about ultrasmall traces and their route to important time savings. So good afternoon, ladies, gentlemen, but more in particular the structures we as human beings are composed of. Welcome organs, cells, organelles, and even the subcellular particles. But first, let me outline the current everyday practice. Face, arm, speech. At what time did it start? Act fast. After a call with the emergency line, the ambulance will take a stroke-suspected patient to the nearest hospital. A CT scan is made, which uses X-rays to make the invisible visible. Is there an obstruction causing an ischemic stroke, or is there a bleeding inside the patient's brain? Although these pathologies are completely different, these patients may show the exact same symptoms. However, treatment methods shouldn't be mixed up. In an hemorrhagic stroke, treatment is focused on controlling the blood pressure and thereby limiting the damage. An obstruction in an ischemic stroke can be dissolved either by using intravenous thrombolysis, or we can enter the vein or artery and remove the clot, which is an endovascular thrombectomy. If we would give thrombolysis to all stroke-suspected patients, we would increase the problem for those having a hemorrhagic stroke. In that case, we're making the bleeding that's already there even worse. And patients that are eligible for an endovascular thrombectomy should be helped in a specialized stroke center, which is a comprehensive stroke center. It might be that a patient needs to be transported to get there. So stroke treatment is all about getting the patient in the right treatment facility within the shortest amount of time. And to do so, we need a diagnosis. And to get the diagnosis, we are currently in need for a CT or MRI scan, which is made in the nearest hospital. And that's why I want to tell you something about extracellular vesicles. These are the subcellular particles that I am interested in. I'd heard of them during my studies, but it wasn't until my PhD trajectory that I got more or less obsessed with them. Extracellular vesicles, both exosomes and microvesicles, are nano-sized structures that are released from all kinds of cells. And they are known to play a role in intercellular communication in diverse cellular processes, such as stroke. And wouldn't it be amazing if we can use these tiny vesicles help diagnosing a worldwide leading cause of disability? So vesicles are actually troublemakers. That's how I would call them. But I hope to make them into problem solvers, because I want to collaborate with them. I want to make it possible that we can already make the diagnosis of stroke inside the ambulance. Moreover, I would like to discriminate between an ischemic and a hemorrhagic stroke, just by using the vesicles that are present in a single drop of blood. You may wonder how I'd like to do that. Well, I would like to improve our understanding of vesicles. I want them to guide me. I want vesicles to reflect the patient's status just by showing me what their composition is. It's more or less like you're in a sports center. You know it's sport since you recognize the building. The type of sport you can derive from the rules they are following and the players that you can see. So if you see teams playing inside, 6-6 players, using a ball and a stick, you might derive it's indoor hockey. Well, the same holds true for stroke. You know it's stroke since you recognize the symptoms. The type of stroke we can derive from the type of vesicles that we can find. So can we find vesicles that originate from an obstruction, such as a thrombus or an emboli? We might conclude that it is an ischemic stroke. However, we might also see vesicles that originate from the brain, thereby originating from a bleeding. In that case, it would be a hemorrhagic stroke. So it's the type and ratio of vesicles that would chose what happened. And I'm looking into that. I'm diving into microliters of plasma to look at the vesicles and look at what the vesicles have caused, which problem they may have caused. Because only if we know the problem, doctors can do their very best to solve it. Yet, I started calling these vesicles troublemakers. Firstly, because they may have a role in causing these large-scale problems, but man, it can be pretty hard to detect them. Because although there are many of them, they are quite small, and there are also many other particles within a blood sample. And we really have to make sure that what we are measuring are actually the vesicles that we are interested in, and not the lipoproteins or shattered cells, broken cells maybe. No, it are really those membrane-enclosed structures that we want to know more of. And once we are able to do so, we have a second problem to tackle: stability. If we would test a stroke-suspected patient twice, we would need to get the same outcome of that test. It's either 2 times ischemic or 2 times hemorrhagic stroke. The same holds true for the measurement of a concentration of a particular vesicle. We want to have the same concentration twice. And after that, we can really look into the profiles the vesicles are projecting. What kind of profiles are they showing us? What kind of neurovascular pathologies are they indicating? Because only if we can discriminate the different kinds of stroke using a single drop of blood, we can actually work on the technique to bring it inside the ambulance. Nowadays, we're using the spacious hospital laboratory to measure these vesicles, but we really have to create a lab-on-a-chip kind of structure. So for now, I would like to wish you already a lovely weekend, and please take a minute to explain this fast test to your friends and family, because it's all of us, we should be able to act fast. And in the meantime, I will keep working on making these tiny vesicles big in decreasing the time until stroke treatment, because after all, time is brain.
Thank you, Naomi. Next up, we will be joined by Anke from the program Neurodegeneration, and she'll be joining us via Zoom.
Thank you for allowing me to present in this way. And on behalf of the program Neurodegeneration, I would like to talk about the selective vulnerability of a really interesting neuron termed the von Economo neurons and its selective vulnerability in BVFTD. What do these animals have in common? They're all highly evolved mammals with an increased sense of social cognition. This is the processing, storing, and applying information about other animals and social situations. Social cognition is recognized to be altered in several diseases, including autism and schizophrenia, but also in a neurodegenerative disease called frontotemporal dementia. Our lab focuses on the behavioral variant of frontotemporal dementia, where alterations in social cognition are seen in behavioral symptoms such as apathy and inappropriate behavior. Underlying changes in neurodegenerative diseases, certain networks are targeted. And underlying these network alterations, there's a microcircuit that has a certain specific type of neuron that is vulnerable. Here we see the pathological progression of the behavioral variant of frontotemporal dementia, or BVFTD. In the end stages, you can see advanced atrophy in the frontal and temporal regions. And in initial stages, there's 2 regions that are selectively vulnerable, the anterior cingulate cortex and the frontoinsular cortex, shown here. These regions harbor a really interesting neuronal type called the von Economo neurons, or VENs, and they're distinguished by other neurons by their shape. They are long, elongated dendrites, and they're found in the mammals with higher social cognition. They're not found in traditional animal models, such as mice and rats. Several labs have already established that they're vulnerable in FTD and also autism and schizophrenia. To find out more about the function, I studied them in detail, and I found that they express a certain marker called GABA subunit theta, or GABAQ. This is a GABA subunit, and it implies that they process inhibitory information in a specific manner. This expression is not found in other regions of the brain, but other neighboring pyramidal neurons do express this GABAQ, which led us to believe that they might be involved in the same processes and that they're related to each other. Here, we aim to show that the VENs and GABAQ expressing neurons are related to each other and vulnerable in BVFTD, And we also try to find more markers that can give us more clues about why they're vulnerable in BVFTD. For this first aim, I included human postmortem tissue of donors with BVFTD, including the distinct pathologies tau, TDP, and FUS. In addition, I also included controls and Alzheimer's disease donors as a kind of relative measurement. For each donor, we counted all the neurons in layer 5 of the anterior cingulate cortex, either GABAQ positive or negative. We then calculated the ratio of GABAQ positive neurons and total neuronal 5 layer population and found that they're vulnerable in TDP and FUS donors, but not in donors with tau pathology. From the clinical files, we extracted the behavioral symptoms, and we found that increased number of behavioral symptoms correlates with the number of expressing neurons in a negative manner. So more behavioral symptoms means less GABAQ expressing neurons, which also indicates that these neurons are indeed related and that they're vulnerable and linked to behavioral symptoms in BVFTD. For the second aim, we used proteomics, where we used frozen tissue from the human brain. We lasered out the vents, and then we processed these neurons further for proteomics, where we can read out the complete proteomic profile. We identified several markers, one of them being DHX9, or RNA helicase A, which is an RNA metabolism protein. And in normal situations, it's expressed in the nucleus. However, in vents and related neurons, it's also expressed in the cytoplasm, indicating that there's something altered in RNA metabolism. When we look at donors with BVFTD, we see a similar kind of expression in the cytoplasm, but much reduced, and also reduced expression in the nucleus. Even here, we can see no expression in the nucleus. So what these proteins have in common with the pathological aggregation is that they're all involved in RNA metabolism. TDP and FUS are involved in describing DNA to RNA, splicing of RNA, and the shuttling of RNA outside of the nucleus to form proteins. In a disease situation, the TDP and FUS are aggregated and these functions are reduced, which also included aberrant RNA splicing and inclusion of cryptic exons. DHX9 is also involved in DNA to RNA transcription and also in protein translation. But what exactly the function is in the cytoplasm, we don't know yet. So here we hypothesized that this aberrant expression of these RNA metabolism proteins can be easily disrupted. So the RNA homeostasis is altered either due to genetic factors or environmental factors, which leads to the inclusion of cryptic exons and also the inclusion of FUS and TDP, eventually leading to neuronal death. So we've shown here that the behavioral changes are linked to the vents and the related neurons, and that they're vulnerable in BVFTD. We also show that these neurons are specialized neurons with an altered expression profile for RNA metabolism, which can lead to the vulnerability of these neurons. So I'd like to thank the following people, especially Priya and Marta. And I'd like to thank you all for your time, and have a nice afternoon.
Thank you, Anke. And finally, for our last Pecha Kucha today, we have Tommy Broeders from Neuroinfection and Inflammation.
Hello, everyone. Thank you for attending my Pecha Kucha presentation. My name is Tommy Broeders. I'm a PhD student at the Department of Anatomy and Neurosciences at Amsterdam UMC, location Rimst. Today, I will be telling you more about dynamic functional network switching in MS and its relation to cognitive impairment. So first about MS. MS is a neuroinflammatory... It's not continuing. So, again, MS. MS is a neuroinflammatory and neurodegenerative disease of the central nervous system that causes neuroaxonal damage, which results in disconnection of the brain. So, you can say MS is a network disorder. Up to 70% of MS patients suffer from cognitive impairment, and network dysfunction plays a key role in this. I think it's not continuing again, but... For normal cognitive functioning, it is dependent on the interplay between different cognitive networks, and dynamic reconfiguration or switching of these networks is essential. Network switching can be quantified using resting state functional MRI, using an approach termed dynamic functional connectivity. So we know there's a strong link between network dysfunction and cognitive impairment in MS. But we do not really know what the role is of dynamic network switching within this relationship. So we would like to know more about this, and we would also like to know more about what happens over time as cognitive impairment progresses. So to study this, we have investigated 230 MS patients and 59 healthy controls that underwent neuropsychological assessment at baseline and after a 5-year follow-up. And they also underwent MRI scanning. Patients were categorized as impaired, mildly impaired, or preserved. And based on longitudinal data, as declining was stable. We applied the sliding window approach, for which the fMRI data was cut up into smaller overlapping windows. And for each window, Pearson's correlation was calculated as a measure of connectivity. And the result is a collection of links between all the different brain regions, signifying how strongly they are connected to one another. Based on that, we could assign each brain region to a network. And for example, you can look at regions 4, 5, and 6. They are highly interconnected. And the same counts for regions 1 and 2. So those would become their own individual networks. And we do this for each window. Then we could quantify network switching using 4 measures. But for the sake of this talk, in light of time, I'm going to focus on cohesion. And this quantifies how many switches a brain region makes from one network to the other, together with another region. And to illustrate this, I've made an example for you. And in the example, you can see 6 brain regions over 5 windows from T1 to T5. And you can see that in the last 2 time points, regions 1 and 4 both switch from the green network to the purple network. And they switch together. So this is what we regard as a mutual switch. So this is an example of a network switching in a healthy control subject. And please pay attention to the frontoparietal network on top and the visual network at the bottom. And you might already see some differences between the network, as the visual network seems to be much more stable than more frontal regions. And also when we look at the group level, we see the same thing, as indicated by the colored points, indicating cohesion for the different networks. On the left, you can see the distribution over all data points, and within that, we represent the mean and confidence interval of global cohesion. And global cohesion represents the cohesion over the whole brain. And what we found is that in cognitively impaired patients, global cohesion is increased compared to the preserved patients. And interestingly, mildly impaired patients show intermediate dynamics, further indicating that with increasing impairment comes more of these mutual switches. So what does this look like in the patient? So here's an example of a preserved and impaired patient. And please pay attention to the sensory motor network in yellow. And again, you might already see some differences between the 2 patients, with the impaired patient showing much more instability, with the preserved patient having a much more stable core of that network. So in extension to our cross-sectional findings, we also looked longitudinally, and what we found is that in cognitively declining patients, you see a longitudinal increase in cohesion over time, which we don't see in cognitively stable patients. So again, this further emphasizes that with more impairment comes more of these mutual switches. But what do these mean? Well, there seems to be some biological relevance to these mutual switches. As we found in healthy controls, contralateral pairs of brain regions show most of these mutual switches. And this is also the case for the healthy control example I've shown you before, with these 2 regions being the 2 that are most often co-switching and also are a contralateral pair. We have found that mutual switches positively associate to MS-related structural damage, such as lesion volume, but negatively relate to cognitive scores. For example, average cognition, but particularly to information processing speed, verbal memory, and working memory. So in sum, we have found the same directionality in baseline and longitudinal analyses, that with more cognitive impairment comes more network switching, and in particular these mutual switches. So then the question is, what do these results mean? Well, the results are in accordance with the seminal hypothesis that states that MS pathology induces a progressive functional network destabilization. But we still do not know why the brain is becoming so chaotic. So there are still some open questions to be answered. Also questions related to, for example, later stages of the disease or earlier stages of the disease, but also related to symptoms such as fatigue, for example, or to treatment. Also, it would be highly interesting to see how the results translate across disorders. Maybe also disorders that are not characterized by structural network damage, psychiatric disorders, for example. And with that, I would like to thank you all for your attention. I would like to thank all my colleagues for their help on this work. Amsterdam Neuroscience for allowing me to present this work to you. Enjoy the rest of the day and the weekend, and please stay safe. Thank you.
Thank you, Tommy.
Yes. Well done, all of you. They're still here, but they put their masks on again, and they were standing here with us all the time, listening to each other. Wonderful. No autocue here, right?
No.
They don't. They just know it by heart.
They know it by heart. They've practiced a lot, and I think they did a really fantastic job, every one of them.
Really, because that was a lot that they told us. Yeah. So what did you think was specific for this segment of Pecha Kuchas? What caught your attention?
Yeah, I just really like to get this really huge overview of so much different kinds of research that is all being done in Amsterdam.
Yeah.
And it's very different kinds, but it all comes down to one thing. We try to understand the brain.
Yes, trying to understand the brain, that is key, that's true. So we're gonna find out what everybody at home or in the office or in the bathtub or the garden or wherever thought of these Pecha Kuchas. And now this is the time where you can start voting. Please go to Slido. In your screen you will see a little cloud where you can click that and you will enter Slido. And if you cannot do that, pick up your phone, go to slido.com, SLIDO.com, and enter the code AM2020. If you were there already, you can start voting right now. How are you? Good, you can only gesture, yes. Is there anyone here who says, well, I think I'll win? Is there anyone here unhappy with how things, apart from the technical stuff? Okay, yeah. Of course, perfectionists. Yes, I thought they were all quite brilliant because
they were great.
So much information in so little time. It's just very special. I am going to take a slow look if the votes are still coming in. They cannot see it. I will hide the screen. No, I cannot see it either. It will be a big surprise for all of us. And let me see, people are still deciding. What criteria do you use when you think of choosing?
I think it's a mix. I think it's, of course, visually what you see, but I think the content is also really important. And I think how the presenters present it, because they're telling you a story and you have to be convinced by the story that they are telling you.
Yes, and this time they also had to handle a camera.
Exactly, yeah.
And talk to a camera like it's a real person. It's exciting, let's see. Yes, I think that we are all... No. Maybe there's a slight delay here. Are you getting nervous already? Yes, are we? We are ready to see the winner. Oh, results. Dirk Jan Ardesch is the winner. Yeah, you can. Oh. Yeah, can I? No, you say something, right?
Yeah, of course, we are all winners, right, here.
You can take off the mask for a second, yes.
These are all winners here.
Yeah.
I mean, this is why we like to do research, to see all these young people doing fantastic research. But we can only give one award.
Yeah.
And
27% of the votes. 27.
27.
So there are many more
that's,
also very popular. Yeah.
So...
I agree. I entirely agree. You know, all winners here. You were already a selection of a selection of a selection, and maybe it was thanks to Priyanka that you are standing here, and one of you has to win, and it's up to the audience. Thanks a lot.
All right.
You have the award, right?
I have the award. It's over there. It's over there, Dirk Jan.
I think I suggest that you can go there and get it.
Yeah.
We'll find a way to make you. Whoa! Bravo! Woo!
Thank you very much.
Yeah, That's wonderful. So thank you all for presenting. And can I please invite the gentlemen to join me at the table? And you, thank you, Priyanka.
Thank you so much
all,
for having us.
all of you. And we'll see you in the future again. Without a mask, I hope.
Yes.
Yes. Bye. Thanks. Yeah.
What a day.
What a day. Mask off. So, what did you think? What did you think of the Pecha Kuchas?
It was exciting.
Yeah?
Yeah.
If you see the Pecha Kuchas of these young people, then I think that's why I'm doing research.
Yes?
Yes, I think so, yeah.
Explain.
Well, I like to work with young people who have good ideas. They are all so smart.
They are.
I mean, some of these stories for me are difficult to understand even, while they explain it very well. And then
You're
I
just
think...
not smart enough.
Well, perhaps. I mean, it's really encouraging to have all these people, young people in your institute. And I think that might also be one of the things that I missed most this year. So normally we are with 600 people, of which 400 are young people.
Yeah.
And they are having discussions about science. I mean, I liked it very much today.
Yeah, what did you think?
Well, I think, you know, they exemplify the real workforce of the institute. They make the hours, and they actually are ready to do team science, you know, much more than we were in the old days.
Yeah, is that the big
I think
difference?
we were trained monolithically. When I was setting up the Masters of Neuroscience and the graduate program many years ago, you know, I thought, gee, I wish I had, you know, followed this master program myself. You know, it was exciting to teach there, and it keeps us going.
Yes. Did you think that team science was reflected enough today?
Today,
Yes.
absolutely.
Today, yeah.
Certainly today.
What did you think?
Well, I thought, you know, in the first session, I thought that, you you saw the interaction already occurring. We have a group,
between the researchers,
we have a group intervention. We call that Young Amsterdam Neuroscience, and this generation should work more together. And that's also what I missed, especially this year. Yeah. That we couldn't do that more intervention between the mid-career team leaders.
Or online versions, I suppose.
Yeah.
Yeah, and that works to some extent.
Of course.
Yeah. But I mean, it's important to mix with people, to speak to people only, well, for 20 seconds to ask, well, how did you do this, or when to do that.
You can go outside and discuss things from a distance in a park, something like that.
Yeah, maybe we should do that.
The summer months it did.
But still, that's difficult. I mean,
true.
And also when you look forward, there will be, well, maybe difficult times coming with the COVID, and people should realize that we are not only here to show science papers or nature papers or good work. We are also here to solve problems.
Yeah.
So people back home should know that they can contact us when there are difficult times and when we can help out.
And reach out. Yes, that's important.
Yeah, I think that's the message that we want to give as the directors.
Yes.
You know, we are there for them.
Yeah.
And so we...
Good to know. You can call them anytime you want.
Absolutely.
At night.
My mobile is in... The mobiles are in our email subscription, the signatures, and so you can always call. There's a low threshold.
Yeah. Please do, yes, because I think that's important too. We're going to ask everyone at home to tell us how important would you rate team science? Can you please go to Slido and please answer that question, because I am curious what people say. Wow. Do you think that they're that polite that they say it's that important, so we're all going to vote a 10?
No.
Because we still have a percentage that says, one, it's not that important.
That's true.
Yeah.
Or maybe people think that that is the highest score. Sometimes that happens as well.
Well, maybe some people don't see the importance of it.
Tell me, what could that be when you say, no, I don't think it's important? And
I don't know. I mean, in my vision, team science is important.
Yeah.
It's not the only thing to do, but I mean, it's really important, and that's how we as directors would like to see the research being done. So,
but a critical mind would say, what could you object to it?
Let me say two things about it. I agree with Diederik. You need teams in order to do the work. You know, for instance, the way in which he organized the COVID biobank just over the last 7 months, that was pure team science. That was his entire lab working 24/7 for a couple of weeks in order to, you know, how many samples? You had many, many samples. And then that was just the start of it. And then that needs to be organized. So science is also hard work sometimes. You have a very clever idea the first 5 minutes on Monday morning. And then it's another 3 weeks of very, very hard work and to motivate your team.
So that could be an objection. It takes a lot of time.
I think the thing that I want to say about that is that sometimes, you know, and there's even papers about that, that real breakthrough sometimes might be an individual achievement. But then I think in these days, in order to have that revelation is one thing, but then you need to validate it. And I think Amsterdam Neuroscience is there to bring good bench research to the bedside, and vice versa. And so you can have a very clever idea at the bench, but eventually the proof is in the eating, so you need to bring it to the bedside.
Yes.
And that's why you need teams.
Proof in the pudding on the bedside. Yes, good. Let's ask another question, because we are curious to find out what you've learned about team science today. And we're gonna, please type your answer, and then we will keep an eye on what people say about this, if they have learned something about it. Somebody told me that she said, look, this person has learned fun. That's good, huh?
There we go.
Today, about team science.
Research is fun, huh?
Yeah. Team science can be fun, probably.
Yeah.
Somebody said it's not really team science when we still put people in the spotlight that have been awarded with individual grants. What do you think of that? I thought that was a nice objection, maybe.
Well, I think in a PhD project, I think, you know, it's like becoming, you know, the next level of being independent, and independence really has to show at the individual level.
All right. So
you can get support, and you can get team support, and you can get into high impact journals with teams.
Okay.
But a graduation is an individual achievement.
Okay. Okay, good. It is. Well, there they say you have to communicate. We've learned a lot about the brains. Yes, we do.
And it's also the responsibility of the principal investigators, which are individual principal investigators, to create team science.
Yes.
So you have these personal grants, but you can use them for team science.
You can.
Yeah,
yes,
of course.
But what does it take to make sure that this is a team effort, except thanking them? Modesty. What do you think about that?
Yeah, modesty. Yeah, you need. That's always good.
Says.
That's always good.
Yeah. So
not only for team science.
And machine learning. That is what we see a lot. That people think that is important and a new development.
Yeah.
Yep.
I think the annual meeting also gives way to wrap your brain on something that's outside your comfort zone. I mean, you know, you don't need to feel reluctant to admit that, you know, you're not comfortable with every discipline.
No.
And we try to provide a safe community in order to learn.
To be honest about that.
That is also team science.
Oh, yeah, it is. Thank you for answering it. Did you see anything? Synergetic impact?
If you have the luck to work within a cooperative team, you might succeed in team science. Yeah, that's true. I mean, it's not always possible.
Not true.
It's essential for future science. I would agree on that one.
Yeah.
You know, there's major projects going on, like major worldwide databases that can only be filled because there's team science.
Yes. Yeah, I can see. Okay, so let me just ask you, what is there left to say about today, first of all? What do you want to...
Well, the experiment worked. You know, it worked in different ways. It was quite a challenge to decide whether we should do a 6-hour session online.
Yeah.
And I think it's also, you know, partly thanks to you that it worked so well.
Yeah. So we are not allowed to thank you. You told us.
No.
But it's true that for us this was an experiment, and I think the experiment worked out quite well. I did notice the PhD students did best, and I tend to agree that, you know, I think the dessert today was very, very good.
Yeah, you think that they did?
It's always the fun part of the annual meeting.
Yeah, and hopefully, also in this poll, you see, hopefully next year, we don't know about the situation, but we can have a face-to-face event again, and maybe we do it in the Amsterdam Arena again.
Yeah.
And maybe we can play a football match, perhaps, on the field. But
there's enough grass there to have many people.
Do it earlier on in the year. Do it outside and meet each other face to face.
Yeah, yes. So because of course people want to meet face to face.
Yeah.
That's healthy that they want to. But also many I see do like the virtual event, like today. Also hybrid. Yes, we can, if it's allowed, if we're all safe, we can. And a smaller face to face event that might be a possibility somewhere soon. I hope.
Yeah, sure, yeah.
Well, let's hope there's light at the end of the tunnel, and let's hope eventually we will meet face to face.
Yeah.
Yes.
I miss that too.
Yeah, of course, everybody. Well, almost everybody does.
Yeah.
Are there other people to thank?
I like today.
Yeah, you like today?
Yeah,
good,
I like
good. Yeah, we were here, we were together, right? That's also different from the viewing part, but I thought this was very good. So, are there people that you want to thank?
Yes, I think, I can thank many people. You thought it was not very wise to thank many people, but I think, I think at least one name pops to my mind, and that is Anita Osinga. She organized this event now for over a decade.
Oh, yeah.
We just celebrated her 10th year anniversary. So she has organized 11 events like this. Wow. This was, I think, her first online event. Thank you very much, Anita and team. And all the others that worked on this in the back and in the front office, the studio here. Thanks to my co-director.
You? Something to add?
No, nothing. I think we will have like a network event, right? You were explaining?
Yeah.
That's important, and I think we had a great day of science.
Good, a great day of science. That is a good conclusion, I think. Well, it's true what you said. We have a network event. We have the themes of the Pecha Kuchas, and they show up in your screen, I think, about now, below. And when we say goodbye, so please stay just a few seconds more, you can click there and then you will be guided to a meeting room where you will find people who chose the same theme. And you can have like a Friday afternoon drink with each other. You have to bring your own beer, wine, or orange juice or water. But you can meet, and that will be nice to find out who you will meet when you hit the button. Everyone at home, in the office, in the hospitals, everywhere, thank you so much for watching and for participating. Read the magazine, I would say, on your behalf. Read the magazine and be great scientists, like you said, for the rest of the century. And maybe we will meet again, face to face. And there will be an event next year, that's for sure. However, there will be another annual meeting 2020 by Amsterdam Neuroscience. Goodbye. Have a great Friday.